Hello, everyone, and welcome back. This is part 2 of the lecture on Chapter 7, Microbial Genetics. We had just finished off in part 1. We had just ended talking about the characteristics of microbial genomes, covering the fact that it's DNAs, double-stranded DNA, and that there's generally one circular chromosome and some smaller plasmids. Now we're moving on into copying the DNA, the replication of DNA. Again, this is something that I'm hoping that you learned the first time, or multiple first times in biology, either in high school or in college. Let's see. So some key features about making copies of DNA is we start out with these parental DNA strands. Remember the double helix? They're complementary strand attached to each other. A's on this side bind to T's on this side. C's would bind to G's, etc. When we say the replication is semi-conservative, that means that these two parental strands are going to stay intact. Even though we pull them apart, they stay connected this way so that the order of the nucleotide stays the same. And then we make a new strand, the daughter strand, we make off of the pattern, if you will, on this parental strand, and then on the other parental strand, we make the complementary strand. This takes energy to make a polymer to put nucleotide monomers together. And the way that it works, previously we've talked about how we use ATP, adenosine triphosphate, to provide the energy to make these reactions happen. Now imagine ATP, adenosine triphosphate, but then there's also GTP, guanine triphosphate, and thymine triphosphate, and which one did I miss? Cytosine triphosphate. So all four of those have these triphosphate groups on, and by taking off two of the phosphate groups, we can release the energy that's needed to make the chemical reactions go. All right, so here's a representation. The original DNA strands, the parental strands, are shown in purple. If we pull those apart and synthesize new strands on each of those, the new strands are represented in blue. Notice that it's half old, half new. Same over here, except the opposite half is old and the opposite half is new. But we kept an old strand. So this is first replication. This is semi-conservative. And once again, when these double helices get pulled apart and then we make copies, the darker color is the original strand and then the lighter color is the new strand. And once again, we can see semi-conservative replication. So it happens each time we pull the DNA apart and make copies. Now, why would you ever need to pull DNA apart to make copies? The only reason a cell would ever have to do this is if the cell is going to divide. So it needs double the genetic information or copies of all the genetic information for the cell to pass it on to the daughter cells. Let's see, I've mentioned the triphosphate. So we've talked forever about ATP, adenosine triphosphate. Here's an example of a nucleotide triphosphate that's not ATP. Here's the guanine base, there's the sugar, and then instead of having one phosphate group on it, it has three phosphate groups on it. That's the trinucleotide. And what it's for is for being able to attach another nucleotide on this strand, on this polymer that's growing. And this is where the energy is coming from, is that the release of those phosphate groups. I'm going to play a video for you that is also embedded in your homework in mastering. So if you watch it here, you won't have to watch it again in the homework. Oops, but apparently I'm not going to do that. Let me see. Let me see if I can share this. Let me try one more time. Starts off a little bit slow.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine.
Okay, so this video is actually for like a general biology class, but they're making the point like it's easier to talk about the bacterial DNA replication, but it's similar to eukaryotic DNA replication. So what we're watching is actually a representation of this happening in prokaryotes.
Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands.
Okay. Origin of replication is a phrase you will hear a lot. It means the place where the DNA first comes apart, where we're going to start copying. And the curious thing is we're going to copy in both directions.
The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them.
Okay, so this is the other important term for you to know. Replication fork, what that's referring to is simply that the double-stranded DNA is pulled apart. And how to represent this, I guess like that. The double strands, if they're held together, the two parental strands, now we're going to pull them apart, and there's going to be this place where we can do some work, but the strands are still connected to each other. And that's what's being shown here.
Many proteins work together at the replication fork.
Oh my goodness. All right, so in blue, we're showing the DNA, and then this thing in green and the things in orange and the things in brown, all of those are representing enzymes. Those are proteins that have a particular shape, and due to that shape, they're able to do some work for us. What do I want to tell you about this? The orange ones are the important players right now, but we're going to talk about some of the different parts of this. And they're kind of anchored together because they kind of work as a team. We have to pull the strands apart, and then we can start making copy.
Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA.
DNA polymerase. So the ACE part tells you it's an enzyme, and then the part that comes before the ACE is the name. It's telling you what the job is. So DNA polymer ACE. It's an enzyme that makes DNA polymer. And there are a few different kinds of DNA polymerases. There are different ones that do slightly different jobs from each other, even within the same cell. The ones we're interested in right now are these orange ones.
DNA polymerase always begins a new strand by adding to an existing RNA primer, shown here in red, that has been constructed first. Original parental DNA strands.
Okay, a few more bits of information that it'll be helpful for you to understand. Think of this like a zipper that you can, start zipping a zipper, but you can't just start anywhere. You can't just like put the teeth together wherever you want. You have to start at that little solid piece of metal has to slip into the other piece so that you can, pull your zipper up. That's what I think about for these red bits. They're RNA primers. They're little strips of RNA. Their function here is like that solid bit of metal to get the zipper started. Come on, where is my play button?
Are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, and DNA polymerase can only add nucleotides in one direction, the new strands must be made in different ways. One new strand, the leading strand, seen here in light blue at the top, is built continuously. The other new strand, the lagging strand, seen here in light blue at the bottom, is built in pieces.
I'm going to give you another analogy to understand that having to build in opposite directions. One is, so you heard the term anti-parallel. As a student, I thought anti-parallel must mean perpendicular, and it doesn't. It just means moving in opposite directions. Think of traffic directions on I-25. You know, you've got northbound traffic and you've got southbound traffic. And so if you were going to, your job were to wash the roads with one of those road washing machines, but you had to drive in the direction that traffic goes, you could get on I-25 and drive north and just continue driving north continuously to Albuquerque and continue cleaning that road in that direction. However, if your home base is Las Cruces and you're going to do this job, you may have to have multiple road washing machines. One gets on at Loman and starts coming south. The next one gets on at 70 and starts driving south. And so you can clean the road in these smaller strips. And that's what's happening with this lagging strand.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time.
Okay, so what's the detail that's not being shown is that if this is an A in dark blue, then this must be a T in light blue. And if this is a G in dark blue, then we have to have a C in light blue. So that's the complementary pairing that's happening. The little sparkle of light, that's representing those phosphate groups coming off, transferring the energy so that we can actually get that connection, that covalent bond to form here. And the enzyme that's helping us with this is in orange, this DNA polymerase.
Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adenine always pairs with thymine. Guanine always pairs with cytosine. Adding new nucleotides works the same way on both the leading and lagging strands.
Okay, then this is like the driving south a little bit at a time on the highway, lagging strand.
Each piece of the lagging strand begins with a short segment of RNA, shown in red.
Like the solid metal of a zipper.
A protein clamps the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase.
And this would be the, get on at the next on-ramp and drive south, get on at the next on-ramp and drive south, little strips at a time.
How are pieces of the lagging strand joined together?
Oh my goodness. Probably here. Sorry about that. Joining the pieces of the lagging strand. Skip it. Okay, joining pieces of the lagging strand. So we're going to get to another solid piece of metal. Now that we've already made that strip of the DNA, that little piece of solid metal, that's no good to us in the zipper. And so we have to get rid of that.
It is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
Oh my goodness. All right, so the different enzymes. So I remember I mentioned that there were multiple DNA polymerases. This is a different DNA polymerase. It still has to bring in DNA nucleotides and put them together, but it has to do it a little bit differently because at the same time it's getting rid of the RNA nucleotide that got put in there to get things started.
How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together.
Okay, finally, DNA ligase is like a little stapler, like to finish that off. It's just one more enzyme that helps us with the copying of the DNA. And why we're going into all of this detail is because later when we start talking about how we combat bacterial infections in the body, in the human body, what we're looking for are, let's see, things that are similar between the processes are very poor targets, because a medication that's going to act on this particular enzyme, if we have exactly the same enzyme, that's going to hurt our cells as well. So what we're looking for as scientists who are interested in this field is, what are enzymes that are different between the bacterial activity and the eukaryotic activity, and then if the enzyme is different in the bacteria, then that can be a target for a medication. It'll affect the bacterial replication, but won't affect the eukaryotic cell replication. That's the goal. So that's why we go into all of this excruciating detail, is to understand what medications act on what, and why some medications have certain side effects that kill off our cells too.
Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
All right, so there you have it. DNA replication, very similar in prokaryotes and eukaryotes. Touching on some of the highlights of that video, we call the origin of replication. That's where the bubble first comes across, and then you can only drive in one direction, which is what they're referring to here. Like, you can only go from 5' to 3'. Not super important, just that you can only go one way. And because the strands run in opposite ways, the new strands have to be replicated differently. The so-called leading strand is the equivalent of driving on the road, where you can just continue driving north, where the lagging strand, you keep having to get back on the highway and drive south, little strips at a time. The DNA polymerases. So here's the original DNA molecule has to be pulled apart. These enzymes that do the pulling apart are helicases. Remember, it's DNA helix. So an enzyme called the helicase is going to pull that helix apart. You have some proteins that have to come into place to kind of keep the strands apart, and then DNA polymerase can do the copying. Yeah, I'm going to keep going. If you need a break, you can always pause the recording.
Enzymes called DNA helicases use energy from ATP to break the hydrogen bonds that hold the two strands of the DNA molecule together, causing the DNA to unzip. The site where DNA helicase unzips the DNA is called the replication fork. As replication continues, these enzymes continue to break hydrogen bonds, and the replication fork moves ahead of the newly synthesized DNA. Stabilizing proteins bind to the single-stranded DNA, holding the strands apart and protecting them from degradation.
Okay, let's move on. One more video.
DNA polymerase can only add bases to the 3' end of the nucleic acid, which means DNA polymerase can only add bases to a nucleic acid. It cannot start replication from scratch. Cells use an enzyme called primase to synthesize a short piece of RNA called an RNA primer. This primer provides a 3' end on which DNA polymerase can act.
So what I wanted to mention here is reminding you that these little strips of red, those are in the analogy of the zipper, this is like that little solid metal piece in the zipper to get things started. And in that analogy, the DNA polymerase acts like that little thing that you hold with your hand and pull up.
An enzyme called DNA polymerase catalyzes the addition of bases to the 3' end of the growing DNA molecule. This reaction is powered by the energy in the high-energy bonds of the nucleotide triphosphates. Hydrogen bonding between the parental strand and the nucleotide triphosphates dictates which base is added. Only thymine fits correctly across from adenine, and only cytosine fits correctly across from guanine. An enzyme called RNase removes the RNA primers, leaving a gap in the DNA which can be filled by DNA polymerase. DNA ligase seals.
Okay, so a number of different enzymes have to take a role in order to remove that RNA primer and then put the DNA in there. And then finally, the little stapler is that DNA ligase.
The maps between the DNA fragments created during synthesis of the lagging strand.
All right. Let's see, a close-up of DNA polymerase doing that, complementary binding here, bringing in the correct nucleotide to make that connection. What else? There's the leading strand can be made into the fork. This is the equivalent of driving north on I-25, and then this is the equivalent of I keep having to get onto the next road and then drive a little bit this way. Somebody else has to get onto the road over here and then drive this way. Little strips at a time. The little strips are referred to as Okazaki fragments. I love saying that word, Okazaki. It's one of the scientists who discovered it.
In many organisms, the two DNA polymerases responsible for replicating the leading and lagging strands are linked together by connector proteins. To make this possible, cells take advantage of the flexibility of DNA. The leading strand is synthesized continuously toward the replication fork in the 5' to 3' direction. Since the leading strand is synthesized continuously, it only needs one RNA primer to start the process. Since DNA
the leading strand, again, one RNA primer to start the process, like think one on-ramp, I'm on it and I can just keep driving. In contrast, the lagging strand down below, you'll see it keeps being made in these short segments.
DNA polymerase can only add nucleotides to the 3' end of the growing DNA molecule. The lagging strand is synthesized discontinuously, away from the replication fork in the 5' to 3' direction. The DNA of the lagging strand is synthesized in pieces called Okazaki fragments, which consist of about 1,000 nucleotides. Each Okazaki fragment requires its own RNA primer.
So repeated of the analogy of the zipper, the little RNA strips are the little metal piece to get things started. In the highway analogy, it's a different on-ramp to get started again. And let's see. The characteristics of bacterial DNA replication, saying here bidirectional, this is not a huge surprise. It happens in eukaryotic DNA replication. Also, you get this origin of replication, the bubble pulls open, and then you get replication in both directions. There are some other enzymes, gyrases and topoisomerases. They remove the supercoils. If you've ever worked with a phone cord, you know how it coils up. If you start like playing around with it, like you end up with this big ball of coil. That happens to the DNA also. And we have enzymes that help us kind of smooth that out, if you will. Think of it like, I don't know, DNA conditioner, like gets the snarls out. The DNA gets methylated. This is, how to explain, adding a methyl group. It happens in eukaryotic genomes also, but it's been discovered that it happens in prokaryotic genomes, and it allows for control of things like expression of the genes, as well as when it's, the copying of the DNA. And here's the origin of replication. You pull the two strands apart and start copying in both directions. And then finally, when you get to the other end of that circle and you've made a copy, you end up with two identical copies of this DNA. Remember that in prokaryotes, often the genome is one big chromosome that's a circle. But the semi-conservative replication, those rules, those still apply. So here's the original thing. There's this dark purple. And then as you pull it apart and make new daughter strands, look at that, semi-conservative, half old, half new. So we've said this before, eukaryotic and bacterial replication is similar. There are some differences. There are some different DNA polymerases. The enzymes are a little bit different, and you start at a bunch of different places. The Okazaki fragments are shorter. And what's a little bit different in eukaryotic cells is the only one of the bases that gets methylated is cytosine. In bacterial, I think it's the adenine that gets methylated. All right, so that is the end of part 2. I know that went long. Thank you for your attention, and you'll be answering questions about this process in the homework for this week. For next week, we will continue with microbial genomics, but we'll cover some other aspects of the DNA and the functioning. See you then. Hope you have a great week.